Okay, for the makeup lab for honors biology enzyme activity, I'm going to ask that you read the introduction and the objectives. And in the introduction, you see that hydrogen peroxide is a substance that breaks down. It breaks down into water and oxygen. And inside this cup, it is slowly breaking down. But it breaks down fast enough that we actually have to purchase hydrogen peroxide in a brown bottle. Otherwise, it breaks down from just even contacting the sunlight, and as molecules move around. And so, so it is breaking down, and we make something like this in our cells, except it's a lot stronger and can be toxic. So we have to be able to break it down. And there is one enzyme that will speed up that reaction so that it breaks down quickly. And that is the enzyme catalase. And so we're going to look at catalase today. It tells you under Part A that you're going to put some hydrogen peroxide in a tube and put some liver in it. I'm going to switch that around, and we're going to do the liver first, just because it's a little easier. We'll put it in the tube and stick it down with a straw. Okay, and we'll add a pipette full of hydrogen peroxide. At the very bottom of this page, you see that there is a rate chart that you can rate your reaction and the bubbles that come out of it, anywhere from 0 to 6. So we'll add the first one here. So I'm going to let you rate that one. And you can put the number in the blank on number 2 beside consider the equation. And then beside the number that you put down what gas is being released. Now, if you'll pick this up and fill it, does it feel warm to the touch? This one feels slightly warm. Not very, very warm, but slightly warm. And if it's feeling warm, that means it's giving off some heat. And so I'll let you decide if we're going to call that endothermic or exothermic. On number 4, you're supposed to pour off this liquid. And they want to know, what is this liquid? What is this liquid we're pouring off? And you might check your equation to help you figure that out for number 4. And now let's add some liver to this liquid. If I can get a little bit more out. Okay, not much. So let's add a little bit more liver to this. I'm going to use a straw to stick that down there. There we go. Nothing happening. Wonder why not. So it says, what do you think will happen? And then the reaction rate you can write down. And so it doesn't seem to be reacting. Maybe you can think of why. And then we're going to take to the first liver that we had. We're going to go ahead and add hydrogen peroxide to that again. And we seem to be having a reaction again. And so now it says, is catalase reusable? What evidence do you have? See if I can pour this off again, and we will be back having just that liver in there. Poured quite a bit of bubbles in here. Oh dear, oh dear. So even though there's bubbles in there, I don't think it came from that liver. Okay, let's take a look at changing the temperature of the enzyme. And it tells us to use some warm liver, and to use liver that's been boiled, and then liver that's been placed in the cold. So we're going to start with liver that's been placed in the cold. A little chunk of that. Put that down a little bit. And I'm going to add a pipette full of hydrogen peroxide here. That looks like it has a pretty good reaction. And then I have some liver that I boiled earlier today. It looks cooked. And so I'm going to add that to a test tube. And then once again, add a pipette full of hydrogen peroxide. Well, it looks like I see some floaty things in there from the liver, but I don't really see any bubbles coming off of that liver. And then I will let you write down what temperature seems to affect the catalase enzyme, and can that effect be reversed. So think about when you put something in the freezer and use it later. Think about when you cook something, and if you want to reverse that, does that work? So those might be some ways to think about that analysis question on part B.